In part 2 of our video series on the functions of membrane proteins, we're going to take a look at a family of membrane proteins called receptors. Now, receptor proteins share a number of common characteristics with both enzymes and with carrier proteins, although as we'll see, the function of these is distinct from both of them. If you don't recognize what the word receptor means in the context of proteins, we can look at receptor as the noun form of the word, while the verb form of the word is to receive. So what exactly are receptor proteins receiving? Well, the short answer is that they are communications receptors, meaning that they receive signals sent by other cells in the form of hormones, and this is where we can see a common feature between receptor proteins, enzymes, and carrier proteins. This is because just like enzymes and carrier proteins, receptor proteins have an active site which is substrate specific, and that means that because of the structure and the polarity of the receptor, only a specific hormone is able to enter the receptor's active site and cause the receptor to affect changes within the inside of the cell. What this means is that only a single hormone molecule is able to turn a receptor protein on or off. Now, that's where the similarity of receptor proteins to carriers and enzymes ends, because the function of a receptor protein is to affect which processes that are occurring within the cell are active and which processes are inactive. Now, the chemical processes that are regulated or controlled by receptor proteins depend on what the hormone is, what the receptor is, and even what type of cell is going on. One of the common functions of receptor proteins is to cause changes with how the DNA inside of the cell's nucleus is working. So remember that even though DNA is the repository of genetic information, it never leaves the nucleus, and DNA needs to be used to produce RNA as an intermediate molecule, which then affects which type of proteins the cell is going to produce. Now, all DNA in the cell is not constantly being used to produce RNA and not constantly being used to produce proteins, because not every cell needs to produce every protein that exists. So one possible role of a receptor protein is to signal to the DNA which sections of the DNA need to become active based on which proteins are needed to be produced by the cell, or which proteins we already have enough of and which proteins we can stop production of. So this is one possible chemical process that is regulated by the binding of hormones to a receptor protein. Now, another function of receptor proteins is actually in cell membrane transportation here. For example, if we have large substrates that can't pass through a carrier or a channel protein in order to get inside the cell, what some receptor proteins do is that they actually signal to the cell membrane itself and trigger movement of the cell membrane in order to absorb certain substances within the cell. And we will talk about this process here in another video. Unfortunately, as we can see in these diagrams, this receptor-mediated absorption of large substances can also include pathogens such as bacteria and viruses that basically trick the cell by looking very similar to certain hormones that trigger receptors to move the cell membrane and let them in. So this is an example of where the function of receptor proteins for absorbing certain substances can actually be exploited by pathogens that make us sick. We're not going to spend too much time talking about enzymes that are found within the cell membrane. Suffice to say that, like all enzymes, these are biological catalysts that speed up chemical reactions within the cell. Now, these chemical reactions are almost always specifically related to the membrane, and very often these enzymes are actually regulated by receptor proteins and by the hormones that bind to them. Now, what this means is that many enzymes that are embedded within the membrane can actually be controlled as to whether they are on or whether they are off, depending on whether a receptor protein has a hormone attached to it or whether that hormone is absent, which gives the cell a greater control over whether the enzymes in its membrane are constantly active or not. The next category of proteins are what we call cell surface marker proteins, which sounds like a mouthful, but their function is actually relatively simple to understand. So you can think of a cell surface marker protein the same way that you think of a passport, a driver's license, or any example of government identification, and that is to identify the cell to the parts of your body that make sure that all the cells in the body are doing what they're supposed to do and that we don't have any intruders like bacteria and viruses that make us sick. So a cell surface marker protein identifies that these cells, in this case red blood cells that we see here, are cells that belong to the human body, and their job is to tell the immune system, that being the system of cells that controls whether your body is fighting against infections or not, and this includes your white blood cells, the primary cells of the immune system, and says to them, hey, we are doing what we are supposed to do. We are cells that belong in the human body. Please do not kill us, because white blood cells are very simplistic in their understanding of what cells should be allowed to survive. If you belong in the body, you can stay alive. If you don't belong in the body, if you are a foreign cell, we are going to kill you, we being the immune system, just to make sure that you aren't doing any harm to the human body. Probably the best example of cell surface marker proteins, let me just write the proper word in here, are actually blood type markers. So for people that don't know, there are 4 primary blood types in humans, O, A, B, and AB, and what blood type you have is dependent on which marker protein exists on the surface of your red blood cells, and the only distinction between them is actually not in the protein itself, it's actually in a oligosaccharide carbohydrate structure that is on the surface of the protein. So very often marker proteins are actually examples of glycoproteins, which we see here. Now, if you receive a donation of the wrong type of blood, let's say that your blood type is A, but you receive B-type blood, your white blood cells will recognize that you have the wrong combination of polysaccharide or oligosaccharide compared to the blood type that you are supposed to be, which leads your white blood cells to kill the blood type that doesn't belong with the rest of your body. For this reason, the technical name of cell surface marker proteins are what we call endogenous. Endo is a prefix that means inside, so endogenous means that they are naturally occurring inside the body, and the word antigen simply refers to any structure that is recognized by the immune system, like an identity friend or foe signal here. The final example of cell membrane proteins is purely used for structural purposes. So unlike all of the other examples of membrane proteins, these don't actually do anything except for contributing to the shape and structure of the cell. These are what we call anchor proteins, which implies that their job is to connect the protein structure to the cell membrane to other parts that exist within the inside of the cell. Now, if you review your cell biology, the complex protein network on the inside of the cell that holds all of the organelles in the proper place and is used to transport substances from one end of the cell to another, this network of proteins is what we call the cytoskeleton. And we can see from this diagram here that cytoskeleton anchor proteins here connect the phospholipid bilayer of the cell membrane to the protein networks that we find on the inside of the cell that make up your cytoskeleton. So part of the function of anchor proteins is to connect to the cytoskeleton in such a way where the cytoskeleton holds the membrane in a very specific shape. And this is what allows different shapes of cells, for example, this nerve cell here, to adopt a completely different structure than another cell with a different function, such as a muscle cell like this. This is all because cytoskeleton anchor proteins connect to the cytoskeleton network of proteins inside of the cell in order to hold the membrane in the specific shape that allows the cell membrane and the cell to take on its specific shape and therefore its specific function. Now that you have finished learning about the different functions of cytoskeleton and cell membrane proteins, you can take a look at this first case study here, which looks at aquaporin, which is a transport protein, and answer the questions about it here. Once you're done that, you can take a look at these practice problems here and see if you can identify the different functions of the different protein components within the cell membrane.